And now we're moving with to discuss the third generation sequencing. Third generation sequencing, very interesting new development in terms of sequencing technologies. Different way to refer to third generation sequencing is by calling them long read sequencing, because this is what characterizes them. Long read sequencing. So it's the newer approach for DNA sequence as compared to the second generation, or what we refer to as short read sequencing. So third generation sequencing are not yet meant to replace the second generation sequencing. These are all developments with efforts to get to read the DNA and again try to achieve high performance and accuracy. When I first heard about sequencing, for some reason I imagined that it's going to be the DNA unfolded somehow and we're going to read it one shot. I didn't imagine that we had to chop it, okay, to read it, okay, and then we have to handle several challenges like solving our puzzle and working with sequence alignment. But this is actually the recent development in the field, which is third generation sequencing. It's capable now of reading thousands to hundreds of thousands of base pairs, as compared to a few hundreds of base pairs for one single read. But a question here that I would like to ask you, and then note it down as a treasure hunt question, is when it comes to short reads, when we looked at the next generation sequencing, Illumina sequencing, and previously Sanger sequencing, why they were only able to handle short reads? Okay, this was a limiting factor inherent in the technology. But why? What was there that made it difficult to go and process long reads? Everyone wanted long reads. That's why third generation is talking about long reads. But it was not possible, giving some limitations. So we discussed different ideas. Not necessarily it was pointed out clearly as that specific limitation that ended up doing short read, but the concepts were there in the modules. So try to think about it, research a little bit, and then share with me your answer for this specific question. So back to third generation sequencing. What's unique about third generation sequencing? Few ideas. Okay, first of all, again, this is like, it's one of the most distinguishing factors, long read sequencing. Everyone is happy about long reads for different reasons. If I can get the full DNA, imagine this, then I don't need to, worry about other steps to come. For example, one of the things that are unique to third generation sequencing, what's called single molecule sequencing, okay? Single molecule sequencing, okay? Single molecule sequencing is one of the features of third generation, unlike the second generation, which required amplification, okay, of DNA fragments. Remember this. Third generation involved the sequencing often of the DNA molecule. So by this, if we can cancel the amplification step, we're actually canceling the amplification bias. So one of the sources of bias is canceled. Not only this, real-time sequencing. Okay, real-time sequencing is another feature related to third generation sequencing. So some platforms can provide real-time sequencing data. And I remember this very interesting visit to the lab where I saw my colleague using the Oxford MinION port, which you're going to see today, it is meant to be a USB, but it's not the USB size perfectly, but you can hook it in the USB style. But it is, you know, slightly bigger than the typical USB. He had an experiment, had the sample prepared, put it there, and here you go. Interesting. He had the flow cell there, and then put the cover on, hook the USB, and then you see real-time sequencing, which is really amazing. Now, SM, this is SMRT. This is why we can call some of these technologies smart, SMRT, single molecule real-time. Now, when we talk about long reads, we can directly come, for a moment, think what steps we can save alignment. And if we don't need to do alignment, then we can reduce our dependence on reference genomes, okay, on reference genomes. We can reduce our dependency on reference genomes with long reads. So some of the well-known third generation sequencing technologies is platforms, Specific Biosciences, PacBio, Smart, Single Molecule Real-Time, as just explained. PacBio comes from the name of the company, Pacific Bio. Okay, so let's dive into this technology, and as always, let's look into some of the pieces or some of the core concepts that makes this technology unique. Okay, so the first thing that we look at, just bear with me. I'm trying to, yes, okay, good. So smart build, so single molecule real-time build template. We have the build shape kind. It is based on two hairpin adapters. So there are these adapters that are attached to sides of the DNA fragments, and together this is going to make what? Circular sequence. This is going to make a circular sequence. So this circular configuration is going to allow the DNA polymerase to continuously read the same molecule multiple times, which is going to enhance the sequencing accuracy. So think now we have DNA, and there are these additional hairpin adapters that are ligated to both sides. And then if you look into this, if we're going to unfold it, right? So you'll find that I can go through this way, right? And then here you go. We're done sequencing this, right? So if the DNA polymerase is attached and working, it's going to keep going. And then here you go again. Okay, it's going to continue. And I'm here, right? So I keep going, and I'm revisiting this again. Right? Very interesting design to come up with this idea. It's a circular design, and again, to force the DNA polymerase to continuously read the same molecule for multiple times, enhancing eventually the sequencing accuracy. So this is one of the concepts here. Another concept is what's referred to as ZMW wells. These are the sites where sequencing takes place. I'm going to see an animation to reflect that, okay? These are the wells here, okay? So they are nanoscale wells. They are at the core of the smart sequencing technology. They are so small and can hold only a single DNA polymerase molecule, okay, and as well as one smart build template that can be sequenced. And what is ZMW? It's ZMW stands for two concepts. ZM, let's do this. ZMW, okay. First we have zero mode. Zero mode, that is the ZM, zero mode, okay. This is just referring to the fact that ZMWs, these wells, are designed to be smaller than the wavelength of the light used for fluorescence detection. So because of their small size at the nanoscale, they have what's called waveguides that are going to restrict the propagation of light photons. So we have these, it's difficult to draw these wells, okay, because they are at a nanoscale, right? And but again, imagine these are the wells here. And inside these wells, we have those hairpins kind of templates, and then there's some DNA polymerase going to read, and kind of they are protective here. So whatever light or photons that are coming out, okay, in a way or another, the mode of light would be protected, as being in these wells. So this is what zero mode refers to, this nanoscale design. And the other term, which is wave, that I've just mentioned, what's called waveguide. Okay, it's a structure that guides waves in general. In the case of ZMWs, wells, they are designed to guide the fluorescence light. Okay, for each molecule that will be read, there's going to be, of course, a fluorescence light. Again, similar to the previous technology that we looked at, a new nucleotide, DNTP, at one time is going to be attached, and then we are going to read the fluorescence, which is cleaved off the molecule. We read it and take an image, and that's it. So the waveguides are designed to guide the fluorescent light, which is, again, produced when nucleotides are incorporated in the sequencing to the detector. So they guide that to the detector. And they are essentially in these nanoscale holes in a metal film that reflects the light. So these are the wells that make up part of this technology, the biotechnology. Of course, there will be, as always, DNTPs. There's no question. We're going to take images, and based on the colors, we're going to have an output. Now, one of the key concepts as well for backbio is the modified polymerase. It's like they got the polymers and they gave it more muscles, okay? The DNA polymerase used in smart sequencing is biochemically modified. It is biochemically modified. Why? To make it more persistent and robust. It's going to continuously keep synthesizing long stretches of DNA without falling off. We don't want it to fall off, okay? This is what's going to allow for generation of long reads. And also another thing about this modified polymerase with some muscles, as the polymerase incorporates fluorescently labeled nucleotide into the growing DNA strand, it pauses momentarily. Okay, again, this pausing is needed for the system to detect the color of the released fluorescent dye. Eventually, we have real-time sequencing output, and see some examples about that. So see here, this is the smart library design. We have adapters at both ends. Okay, it's going to allow for the circular read of the circular reads of the DNA. Now, what we see here is that some steps that comes before, preparing the sample here, eventually going to the machine and to the sequencing. Before the DNA can be ligated to the adapters to form the smart bill, okay, before the DNA goes to this stage, it is essential that the DNA ends are clean and compatible for ligation, because we have these hairpin kind of sequences that are going to be ligated to the size of DNA. And the DNA repair step also ensures that the fragmented DNA has blunt ends, and that any damaged bases or other irregularities there are corrected. Okay, and this is usually achieved using some enzymes and mixes of enzymes that biotechnology technicians would be studying. Then there will be adapter ligation. Adapter ligation, so once the DNA is repaired, it is ready to be ligated to the smart bill adapters. And these are, the adapters themselves are special double-stranded DNA sequences, with the hairpin loops at both sides. And this is what's going to make the whole DNA circular and so on and so forth. And after the adaptation ligation step, any remaining linear DNA and free adapters are removed. So, in summary, this is going to go through certain steps, look into eventually having the SmartBill library of sequences ready, and then things would go to sequencing. We have to attach adapters. We understand this through the experiments. And here you go. I think this is really nice animation. Do you see these nanoscale areas? This is now DNA polymerase. You see the once every nucleotide is attached, you see the wavelength is going to come. The machine is going to read that. You see at this scale, there's one DNA polymerase, one SmartBill. There are millions of those. See, again, it is this what we call or refer to as ZMWs. ZMW is cells of with millions that contains millions of these cells or wells of zero mode waveguides. So they are deeply protecting the wave. Okay, and so eventually through reading the parts in a circular way, I get this information read and it's going to be sent, and then based on the light intensity, we'll be able to tell that an A, C, T, or G. Okay. So for looking into one of the ideas as well in back biotechnology is you start with high quality double DNA stranded, there is ligated SmartBill, and so on and so forth. Anneal primers, DNA polymerase, get the circular DNA. We'll end up with reads, right? Reads, okay, group of reads. Those eventually are going to generate what's called hi-fi read, which has more than 99% accuracy. The consensus of these reads, after we remove these adapters, the consensus of those, once we align them together and take their consensus, will give us what's called hi-fi reads. And these reads, again, are generated using the process that we refer to as circular consensus sequences. Okay, and hi-fi reads achieve accuracy levels comparable to the traditional short-read sequencing methods, and often exceeding the 99.9%. So this summarizes some statistics about these technologies. You can see BacBio is the only sequencing technology now to offer not only long reads, but highly accurate. We look into nanopore technology. It is able to achieve long reads, but not such high accuracy, okay? Sanger reads are more at this end, okay? It is high accuracy there, but small read length, okay, or short read length, okay. And long reads also, BacBio has developed this and keep developing this. So now you can refer to hi-fi as the state-of-the-art, very accurate long reads. So when we have very accurate long reads, if we have very accurate long reads, think about a group of patients or a group of species. What could be the applications that we can take advantage of if we have long reads, okay? Short read sequencing, okay, are sometimes, or not sometimes, they are very short, okay? Short to detect structural variations. So I'm gonna consider this a hint and give you a moment to write down your answer for the treasure hunt question. Now we can go with long reads. What could be the advantage of this? think about few applications or one application. So when we are having the long reads, we can look into what's called structural variations or structural variants. There are many types of variants as we look here. We looked before into insertions, deletions, and structural variants, insertions, deletions, and these would be less than 50 base pairs. Okay, less than 50 base pairs is just bear with me a second. This would be less than 50 base pairs per one read, per one single read, which is, in terms of what levels of information we have access to make a comparison between a group of species or so is limited. Okay, the short reads are too short to detect more than 70% of human genome structural variation of human genome structural variation. Okay, human genome structural variation. So this we have single nucleotide variants, right? One base pairs. We have insertions, deletions, less than 50 base pairs. Short reads are short in hundreds. Structural variations are usually longer than 50 base pairs. And there are different structural variations when it comes to understanding the species. Could be deletions, okay, between, having two human, okay. Could be deletions if we're looking at two genomes trying to gain insights. Deletion of a segment. For example, the deletion of segment within the dystrophin gene can lead to what's called Duchenne muscular dystrophy, one of the diseases. Insertion, a segment of DNA is added to the genome, and this also could have an effect. For example, the case of hemophilia. All these diseases are really major. Duplications, inversions, translocations, copy number variations. Many of these types of structural variations will be more can be performed in a more systematic way once we have access to longer reads. So this is an example to show the alignment of several assembled genomes, and then eventually trying to link this to gene expression. Somehow we're trying to go from genotype to phenotype in this case. So having long reads, of course, and a way to access such information that we have structural variance types is going to be very helpful in understanding the population aspect. And so if we have two groups, like in this case, and we're using the heat map here, a kind of visualization, graphical visualization to show the intensity of genes. It's an ordered set of genes. And then we're trying to link this for each set of genes, if there has been, or parts of the genome, there has been some insertions, deletions, and try to really group these together. Of course, we need a source to have this information. And so with longer read sequencing, this would be more accessible. This shows for smart cells of 8 million and smart sequencing applications. What are the estimated costs in US dollars per sample? If it is this de novo assembly up to 2 gigabytes, we're looking at $1,300, okay? And looking into whole transcriptome, $1,300, okay? Shotgun metagenomic assembly, $1,300, and so on and so forth. So it depends on your needs and experiments, and you can refer to this for further, information. So this covers one of the technologies that we're looking into here. Give me a second to close the curtains. Okay, sorry. So now to nanopores, it's amazing. When I hear about this, it's a USB. Like this was what? Can we imagine the future? We have smartphones, integrated devices, and then we have our personalized assistant, personalized medicine assistant. I think this may happen in the future. Okay. I would say may, not going to happen because still there's some sample preparation, but who knows? Things could scale up and then we end up many things integrated. But again, to which extent we believe that the patients are going to pick up their own medicine and how AI is going to replace experts in the field, this is all up to some philosophical discussions. If you are interested, we can have those in the office hours. But again, it's such amazing development of what, couple of $1,000s. We can get now a full sequencing machine. Who imagined this would happen? I'm not talking about the cost of one sequencing one genome, no, or one sample. I'm speaking about the cost per the sequencing machine itself. So you can visit Oxford Nanopore website, see the pricing is unbelievably accessible now. And I have seen one of those. Very interesting, very fun to look at and play with. Okay, and again they have what makes them unique. So Oxford Nanopores Technologies, again the name of the company. That's why we refer to them as ONT. Okay, ONT, Oxford Nanopore Technologies. It developed this technique, and it's really an innovative DNA sequencing. I don't know, believe it or not, we're not gonna use fluorophores here. Wow, so this is gonna be something new. Okay, for the first time, electricity is gonna be used. Okay, we'll see how it's gonna work. So unlike other sequencing methods that are based on fluorescence detection, these ones read by DNA by monitoring changes in the electrical current. Okay, this part here from positive to negative. Okay, as DNA molecules pass through a nanopore. And a nanopore, this is a nanopore is a tiny hole. And this is typically formed in a synthetic membrane or a biological protein. Oxford Nanopores uses protein nanopores embedded in electrical resistance synthetic membrane. So nanopore is one of the things that make up the Oxford Nanopore device. Okay, this is a highlight for that part. Okay, and in terms of sequencing mechanism, as the DNA or RNA molecule is threaded through the nanopore, each nucleotide causes a characteristic disruption in the current. Each nucleotide would have some signature. By monitoring these changes, we can tell which one is which. So some of the key concepts that we want to go through here is the design of the adapter or the template here. Okay, now we don't have two hairpins here. Okay, this is the leader hairpin template. So there is one hairpin sequence here. So it's a different structure than compared to PacBio. And this hairpin adapter allows the complementary strand to be sequenced immediately after the original strands, because again, we are going this way, right? We go here and immediately we go to the opposite or reverse strand. So this kind of provides two-directional read from a single molecule. Okay, so this is the template design. We can now have bidirectional sequencing because of this. So keep going, and then it's going to end up reading the other part. Now look into this part here. We have the following. We have the motor protein, and we have what's called alpha hemolysin. Okay, I hope I'm pronouncing it right. Okay, if you are a chemist watching this lecture, please correct me if I made a mistake. So alpha hemolysin or hemolysin, this is a protein that forms the actual nanopore in the membrane of the MinION device. This is the one that forms the actual. It is a large biological pore, and it's capable of sensing the DNA. As the DNA or RNA passes through it, okay, changes in the ionic current are detected. Changes in the ionic current are detected, which correspond to the sequences of the nucleotide. What is the other part, which is a crucial component of the sequencing process? It is the motor protein that we see here. This motor protein, it controls the speed at which the DNA or RNA is threaded through the nanopore. So the speed needs to be controlled. If we feed this directly, the speed might not be controlled, causing some bias or issues. So by controlling the rate of translocation, in this case, the motor protein ensures the nucleotides are read with precision. And also, for DNA sequencing, the motor protein also unwinds double-stranded DNA, allowing each strand to be read sequentially. Okay, you see? Unwinds. So we have sequentially. Eventually, we have the ONT output. ONT, the Oxford nanopores output. There are current shifts as DNA translocates through the different pores. Usually it's depicted with these squiggles output plot. And we try to then detect if these were, any of the 4 nucleotides. Now, this is what? Now, here's it's just for fun. So another treasure hunt question. You have seen this before for many, many times. Okay. Now I'm going to give you through the course page. Do you remember where? Okay, I'm going to give you a moment to try to remember. Okay, and then share it with us before I go to the animation next, where you will know where you have seen this before. Okay, this is actually the GIF figure used for GIF figures used for the course icon. Okay, so let's see this. See these attachment going to happen here, and this is the blue part, the nanopore. The motor protein, it's the purplish one. It's going to bring this here, unwind, and then send the DNA strand, and then molecule by molecule, the electrical current will be detected. Yes, this is the one. Now we see the electrical current coming here. Okay, we have, this is the output. Okay, the output we don't know yet. Before we have the output as green, red, blue, green, red, blue, or yellow, whatever, where the greens at fluorescence dyes that were telling us ACGT. Now this is what we have is a signal, So we need to do something, usually some modeling, okay, to try to detect the sequence coming from the current. So if you look into it, part of the modeling that is done is building what's known as recurrent neural networks, and these kind of neural networks with layers that where the output is fed in the input again. Okay, these are the models, the standard models used for sequential data. Because part of the processing here, we don't want to process this independently from the next nucleotide. There is some relationship as we go through these in terms of signals. So this will be parameters learned from training data. So there are some models that are fed with these signals. If features are extracted, and then through layers of what's called or unique components of what's called bidirectional information to make sure that the information is flowing both directions to build the relationship of the sequential information and not assume every nucleotide is independent, and eventually be able to decode the sequence and know if this is A, C, G, or T. And this is more in-depth illustration of what is used in that recurrent neural networks with long short-term memory architectures. These are key concepts, and if you are more into this, We can also dive into this. It's under the umbrella of machine learning and deep learning. Language models, for example, are using some of these ideas to try to build a relationship between the characters and the words in the text. So long reads there, some models or functions, let's call them functions, are going to capture the relationships and eventually give me the output information and perhaps summarize some modification in the genomic position of interest. So with that, we have looked into some of the third generation sequencing. We're going to move now to FASTQ file format again, and have a recap, because this is what we want to focus on at this stage. After looking at the sequencing, this is what we're going to work a little bit more with. Okay, so we looked into the library preparation, right? There's cluster growth when we visited the Illumina sequencing, and then we have base calling, image acquisition. Eventually, we have a set file. Okay, now one of the things to note here is reading the statement. Different experimental sequencing methods may cause different biases in the data. So something we have to understand. Bias is there. Every sequencing technology is going to have its own bias. We have tried to highlight some, and there are much into this, depending on the technology. When you're going to use one of the technologies, it's very important you ask about the bias. I remember I had this interview with one of the top pharma companies in the States, and they had a very interesting technology. I was amazed when they introduced me to the technology, but yet they had lots of noise. So sometimes, especially at the beginning of a new technology that's helping me to scale up and do millions of experiments, there can be a caveat there, okay? There can be some bias, so we have to be careful. So that's why we have to look into the quality information. FASTQ files have and holds this information. Each sequence that we have here, we discussed this before, we have the sequence header information, giving me the locations and different information. Then we have the sequence itself, and then this is a separator, and here is the quality. Here's the quality is indicated with the ASCII score. It is represented as an ASCII character. Okay, with the actual score being the ASCII value. So each character, you can go and get the ASCII value for this, okay, the ASCII value. And then once you get this value, subtract from it 33. That's why we called it Fred or Fred score. This is how we get Fred score, okay, or what we call Fred plus 33 encoding, okay. So, for example, if the ASCII character here is, J, we don't have J here, but we could have J, okay, so let me actually put it here for clarity. If the ASCII character was J, it corresponds to a Fred quality score of 41, okay, and so on and so forth. Now, how many sequences reads are represented this portion? So this is a question for you. Note it down as part of the treasure hunt questions. Now, as we move here, we're trying to know what is the quality, okay? Now, one thing that we have looked into, I've mentioned before repeatedly, this is not one quality score. This is the quality for that specific nucleotide in that sequence. Okay, this is the quality score for that specific sequence. For example, nucleotide like base T here, the quality, if we decode it, is going to be read as 25. Okay. Now, typically a Fred score of 20, which corresponds to an error probability of 1%, is considered a threshold of acceptable quality. Okay, this is gonna mean we have 99% of accuracy. If 20, a score of 30 of error is considered high quality, which is gonna mean 90.9%. Okay, that's the score of 30. That's the score of 20. So by looking into this, is the Fred quality score represented. And it's going to give me the probability of accurate, how accurate is this, and maybe the chance that there is an error. So a question that we have here for looking into this, is this read poor or good quality? Now, I don't want you to run into the exercise. It's up to you to check for each one what is the output quality score. But I want you to share with me an idea. If I end up having as a reference, you can see the reference here for the ASC quality encoding, okay, with all the range. So what is this? Is a colon, okay, this is a colon, so it's going to correspond, I guess, to 25, okay, and so on and so forth. Whatever, let's take anything. For example, I is going to correspond to 40. So we'll end up with a set of numbers for each single nucleotide. This is cytosine here, is with very high chance, very high confidence, with a high probability that this is correct, cytosine, okay, as compared to very low probability in the sequence that we're looking at here, okay? So what could be an idea if we have a set of numbers to come up and find the quality of the full read? This is one read now. We have maybe, I don't know, what is the length of this? 100 base pairs, 300 base pairs. We'll get 300 quality scores for each nucleotide. So here's my question for you, which is, how can we get quality information for the whole read, not the single nucleotide? So this is a reference table for the base call accuracy. So we're left with one activity. I'm going to share more information with you in this regard very soon. Thank you so much.